For those of you that don't know, my lab is at Imperial College in the Synthetic Biology Center there, and we've been set up for about just over 2 years since our labs opened at Imperial in the Synthetic Biology Center. My group work on synthetic biology and starting to work now on genome engineering as well, which I'll discuss a bit in this talk. And we work mostly with classic model organisms, Cerevisiae and E. coli, as well as having a collaboration with British company TMO Renewables to work with thermophile chassis as well. So everyone seems to have had their definition of synthetic biology so far in this talk. I'll just throw in that the way it sort of comes to me is effectively from these two research papers that sort of defined the start of synthetic biology in 2000, which were the toggle switch paper we see here from Jim Collins's group, and the Repression paper that Michael Elowitz was the head author on. And what you'll see from these sort of figures, it's illustrative. You're seeing modular parts coming together, and you're seeing the fact that you have several parts and control elements involved. It becomes a combinatorial problem, like Doug Kell was saying yesterday, and that means then you have to start using mathematical modeling to start being able to inform and explore all the space that you could possibly design within these systems. So these first synthetic biology projects, the Toggle and the Repression, give a good example of what typical synthetic biology projects are, which I see as apps. So these are apps that run within an existing operating system of a cell. And so you could think of perhaps an iPhone as the cell, and we design and build apps to go into that to perform new functions. And actually in the Toggle paper in 2000, it was actually Tim Gardner and Jim Collins suggested that what they were running was an applet, a biological applet. At the time, I think applet was a phrase used for people writing in Java, so you'd have a Java program running inside, in those days, I guess, Netscape Navigator. But it's funny to see how much applet has changed to app, and now it's something everyone knows. So at the parts-based level, the synthetic biology we do is effectively app creation. And we do this following the engineering paradigm that Dick Kipney covered yesterday. So in this talk, I'll talk a bit about our work on app-scale synthetic biology and run through a few examples. And then I'll look at how we're now trying to look towards OS-scale synthetic biology, which is to work on the genome and to see where the challenges and opportunities are going to be to bring app-scale to OS-scale. So back in, I guess, about 2007, 2008, I was a postdoc at Boston University with Jim Collins. And we had this publication which came out April 2009 in Nature Biotech, which I'll talk through a little here. And this was the idea of using models to help guide the design of the synthetic gene networks, these sort of apps that we were working with. And this was done in Yeast. And we set up this sort of forward engineering paradigm here to try and address this issue, which at a talk when he came to our center, Ron Weiss said, he gave an anecdote of a gene network that he'd come up with in his head on an airplane and drawn it all out and thought about it and thought what parts could be used. It took him 3 hours to think about it on the airplane. It took his team 3 weeks to make a nice model of it to predict how it behaved, but then it took 3 years to actually build and get the network functioning in cells. And Ron was quite eloquent in describing all the issues in getting a network functioning, and it was very much that initial networks just kept failing because there weren't enough parts and the parts weren't the correct parts, and then you would spend years going through this cycle of tweaking the parts so that they would fit the parameters required by the model. So the issues in 2006, 2007, when we came to this, the issues that we wanted to address was that there's not enough well-characterized parts so that the modeling that you do is accurate and can really reflect what you're going to go and build. And that the available parts that people pick and choose, particularly things such as promoters and transcription factors, they differ in all sorts of multiple parameters. So something like in E. coli, a P-bad promoter is a typical one used in synthetic biology, and then the P-lac promoter, they're two regulated promoters, but their whole architecture, the way they're repressed, the way they're induced is completely different. So there's all sorts of different parameters coming into play here. So we turned and looked into the literature and dug up this method from DTU Denmark for creating synthetic promoter libraries. This gives you a method to effectively generate a whole range of promoters. And you use this with the diversity added during the synthesis. This gives a sort of fast and simple generation of a part library for your synthetic biology. So what we're doing here is we're kind of doing a lot of what you see in synthetic biology, where you do diversity, mutation, and screening, but we're not doing it on the whole system or on the whole cell. We're doing it very specifically targeted to parts where we know that what we need is we need a whole range of outputs of those parts. How this works is quite straightforward. You target the diversity to the non-critical regions of the DNA sequence of the promoter, and the sort of local changes either side of a key site in terms of the behavior of the DNA double helix then affect how efficient that site is, and then effectively give you different outputs, in this case for the promoters. And what we really like about this method is, by and large, what you generate is the promoters that have a different output, but all the other parameters around them stay exactly the same. So for modeling, that's great. That means we can have a part where we've characterized that part perhaps in the past, something like the PBAD promoter system, and then we can do mutation to have a whole range of different outputs, but all the other parameters of that part stay the same. So with this, we adapted the SPL method to generate regulated GAL1-based yeast promoters, and we selected a library of 21 TET-R regulated ones, so we've engineered in regulation here, and 21 lac regulated promoters. And these effectively now are our nuts and bolts to help us put together the apps that we want to build for synthetic biology, whether they be, in our case, synthetic gene networks, but also these could be used to help tune metabolic pathways. You might have a metabolic pathway with 5 or 6 genes required, so you'll need 5 or 6 promoters of different strength. And the thing we added to the original SPL method is that by also having diversity in the sequence alongside the operating, well, the regions where TET-R and LAC-I bind, you affect the efficiency of how well TET-R and LAC-I repress the system. So you're starting to see differences. It's quite hard here to see on a linear scale, but you're seeing differences in the input if you consider the synthetic, the regulated promoter as an input-output system. So you're seeing a different efficiency in the repressibility of these promoters as well as their output. So this was, I slipped this slide in based on the comments yesterday. This was an example with our first promoter library of the first network we went and built, which was a feed-forward loop network. We'd previously worked in other systems with TET-R, LAC-I, and these 2 promoters here, characterizing them in different contexts in yeast. And we used the data, or Zhao Wang, who was the modeler in Jim Collins's group that I worked with, used the data from those previous experiments with the data that had been captured by our characterization here, and wrote a model of how these feed-forward loop networks would behave, having never actually generated one of these networks. And we realized that the behavior of the network could considerably change by changing the member of the library we put into this system. So that's effectively, we can select from this library here, and because we have different input and output in those regulated promoters, we get different behavior of the whole network. And what we see down here is actually then we went and built several of these, and they match reasonably well the model profile. And although you can see it's not a perfect match, for synthetic biology to be able to have something that works first time very close to what the model was predicting is a major achievement. That can't unfortunately be done for everything. So this second network we went for, the timer network, relies on a temporal change. So that is effectively, we set off an induction, which is either tetracycline or doxycycline, which attacks this. I guess this is kind of like a leaky toggle switch here. So with the induction, you see the flipping of the toggle switch, but then as it leaks over time, you get this sort of timer effect where over a specific amount of time, the toggle will leak back to its standing state. We tie this to GFP to be able to read an output. And we did not know just from previous characterization data everything required to understand what this reset time was. So the paradigm we follow here is we make an initial single version of this network, effectively a prototype. We do the experiments to characterize the reset time, and then we can then extrapolate from that one experiment what happens if we change the promoters for other members of the library in both this position, and also potentially in this position as well. So we're looking now at over 16,000 possible variants because we have 21 options in each of those positions. So for that, model-guided design is absolutely essential. And the reset times are predicted here. The two blue lines are sort of the upper bound and lower bound in terms of the confidence, the area of confidence that Zhao has modeled, and then the red dot is the actual reset time that was measured when we built that combination of timer based from our library promoters. So a pretty good match there. And we decided to not just have a nice little app that did GFP, but actually try and tie it into something useful and something that the yeast cells do naturally. And that is the phenotype of flocculation, which when the cells flocculate, they also sediment. And this is the sort of phenotype you see in beer production, where white beers and Hefeweizens, they have a low amount of flocculation. They have the yeast still sort of floating around during the process, whereas lagers tend to either have very strong flocculation, or the actual companies will go as far as filtering or centrifuging large amounts of beer to get rid of the yeast. So our timer network can easily be attached to a flocculation gene, and we can get predictable timing of flocculation from our networks here. So we have some that flocculate after between 60 and 72 hours, and then you get your sediment there, and others which last almost a week before they then sediment. So moving on from this now, in my own group, we're looking at extending this paradigm further. And our idea was, when we were doing that work in Jim Collins's group, we were working with Gal1-based libraries. So these promoter libraries were still heavily dependent on existing things within the cell. They're dependent on galactose-glucose regulation. And in terms of parts that could be reused by lots of people, perhaps maybe you have like 10 of these promoters in a row controlling a 10-gene metabolic network. That's a bit of an issue because these parts are relatively long, about 600 or 700 base pairs to get the whole Gal1-based promoter. And if you have large amounts of homology in yeast, the native recombination systems of Saccharomyces cerevisiae will match up those same sequences and perhaps delete anything in between. So we instead wanted to try and do this in a more professional manner and take a short, simple backbone promoter and start using this same sort of idea of diversification in targeted places to take this single promoter and create it into a whole range of promoters useful for synthetic biology in yeast. So first off, we turned to existing data. Ben, in my group, scanned available bioinformatics data from many microarray experiments, trying to find and identify very stable yeast promoters that in all sorts of microarray expression sets, so perhaps what happens in a chemostat, what happens when you change the carbon source or you change the temperature, the mRNA profile of certain genes stays the same, and these would be candidates for naturally constitutive promoters. So it was interesting. We sent a few emails out to some experts in this, and we said, we have this project. We're looking for the most boring set of data you can find, right? We want the thing that never, ever changes, whatever. And we got some nice candidates here where really there's very little change. We cloned some of them, 4 against ADH1 and Psych1, which are commonly used. And we tested ourselves, so not microarray, but this time GFP expression with flow cytometry as well to see how the single cell variability is. And from that, we had these candidates, and we went for PFY1P, which had a pretty stable profile and a low cell-to-cell variability, and crucially also it's a very small promoter. So PFY1P has been studied briefly before. There's some papers out about the turn of the century. And this is an interesting, well, for us it's an interesting promoter, but I guess for everyone else it's a very boring promoter. It has no tartar box, it just has a poly-DT stretch here, which causes the bend in the DNA, and the only transcription factor that binds is Reb1p here. And effectively what everyone seems to think is the Reb1p just keeps this as a nucleosome-free region so that the polymerase can just load up time and time again and just keep firing the promoter. So that's nice for us, very simple. Not many parameters will get messed around with if we mess around with this DNA sequence here in this core region. So that's precisely what we did. We went back and applied that mutagenesis strategy again within that core region. And we were able to generate a nice library pretty quickly, 36 members in this case, of constitutive output and with a graded range. And this will be useful particularly for metabolic engineering projects where you might need to tune several enzymes for balancing flux. We wanted to go further than this. We wanted to add regulation because for the sort of synthetic biology networks we work with, these apps that we're going to run inside Yeast, regulation is gene regulation is a key part of that. That's the sort of wire that links things together. So we were able to add the sequence for the bacterial repressor TET-R again, similar to how we did back in the 2009 paper. And the binding of this to the core promoter region blocked the transcription from starting. And this then allowed us to generate, I guess, what is an inverter or a NOT gate, something that's tunable with anhydrotetracycline or doxycycline. So this idea of introducing regulation is really important in synthetic biology, especially when you come from the angle of synthetic biology where you're comparing it to computation and rewiring and having NOT gates and logic gates. This sort of idea of the cell as being something that we can reprogram like an Arduino circuit here. Actually, the cell is much more complex. It has an incredibly wired up network, just like this exchange box that is out on the street in London that I saw them opening up and fixing. There's a huge mass of wires. That I guess is much more like what it would be like to peer in and look at all the transcription factor connections in a natural yeast. So to avoid crosstalking with all of this, when we stick our networks in, it's really important to have non-native wires, I think. And LAC-I and TET-R coming from bacterial systems are examples of ones that we can then use in yeast with some sort of confidence that they don't interact with the native system. The problem here is there's very few of those available, and that really limits design complexity. I think once we've run out of LAC-I and TET-R, we're starting to look around, and there's not much else. There's a few phage transcription factors, but we're going to run out pretty soon. So to address this, we switched and did some, I guess, very naive protein engineering with this new fantastic type of transcription factor, TAL effectors. So many of you will have heard of TAL effectors, and the big push for TAL effectors has been tail ends, customizable nucleases, and these TAL effectors, which wrap around the DNA like this, have a sort of an understood and translated code now of which protein motifs bind to which bases of DNA, so that you can shuffle around these 4 motifs so that they bind to the 4 different DNA base pairs. Effectively now there's several kits, and we ordered one of these from AdGene, that allow you to do a bit of DNA assembly in the lab. Maybe it takes a couple of weeks, and you've created a tail or a tail end that is specific for anywhere between 15 and 35 base pairs of DNA sequence of your choice. And there's some nice online tools to help you design these. So we sort of hacked the kit that we got from AdGene and took out the nuclease part of it and took out the activator part of it that works in plants and mammalian cells, and just had it so that it's essentially the DNA binding domain plus some essential parts either side, so that effectively just generating this core part of the protein that just binds the DNA. And we have this, we've called this a TALOR, TAL orthogonal repressor, because if you bind it directly to the core promoter region, as we've done with the tetracycline, the TET-R system, you can inhibit transcription from starting. So that's exactly what we've done here. We have the tailor binding to the core promoter region, and the great thing about this is with a tailor that is perhaps a motif of 19 base recognition, we effectively have 4 to the 19 possibilities that we can engineer from our kit. Our promoter library has a 4 to the 48 size because we're mutating 48 of the a theoretical size of the library. Obviously, we won't go and screen all of those. That's quite a lot of work. The sequencing costs would be quite high. But the theoretical size of the promoter library we can generate with our oligo method is 4 to the 48. So you can see you could generate, in theory, 4 to the 19 different pairs of a Taylor binding to a promoter just by starting from one promoter and one sort of Taylor architecture. So this sort of bursts open that problem of there not being enough orthogonal regulators in yeast. We now have a schematic where we could easily just keep mutating promoters and keep shuffling around the sequences of the Taylors so that we have orthogonal pairs. And we've done that here in a few cases, and we see orthogonality in terms of the repression here. So this work, this PLOS1 paper that was out this year, we're now putting this to task to start generating some of these second-generation synthetic biology devices that were highlighted in 2009 by my old lab. The sort of networks like adaptive learning networks that require actually quite a lot of different repressors that are orthogonal to one another and multiple toggle switches like in this intelligent navigating biosensor system here. And libraries are characterized biological parts, as you heard from Dick, as an important part of what we do at Imperial College. So we've been working with Biofab, who have been doing this excellent work using similar techniques as I've described today for E. coli part libraries, and we've partnered with them and to make our own data sheets that Dick showed you yesterday, giving the high quality data and metadata for a small number of those parts, and partnering now with TMO Renewables to generate similar promoter libraries and part characterization for their thermophile chassis. We're also starting to work with some new characterization technologies. This is not published yet, but just to give you a hint of what might come, we're looking now at live measurements of RNA levels using this spinach RNA motif that the Jaffrey lab reported last summer, which allows you to have fluorescence measurement in E. coli based on the presence of the RNA. So it's a mimic of GFP, but it's actually an RNA structure that's useful for us in terms of characterizing the different control parts of gene expression. And with Guibart Stan and the student Rhys Algar, we're starting to look at measuring the cost a device running within a cell has to the cell. And we can see that for example, when we turn on gene expression, we can see huge decreases in the amount of free ribosomes available in the cell, in the similar sort of paradigm to have a badly wired up house. When you turn on the tumble dryer, you'll see the light ahead of you dim. We're measuring in a similar way to that to be able to see how gene expression capacity is affected by synthetic biology. Okay, so now to move on to the OS scale stuff. I really like this picture. It's a lot of fun. So what we're thinking now for the next few years in my lab is how do we link genome synthesis work, such as that of the Venter Institute, with the sort of parts-based synthetic biology and the part scale, app scale synthetic biology we've been working on. What would it require to go from these parts to then build up an entire genome? This is a pretty grand challenge for synthetic biology, but you'll also learn a lot along the way in terms of understanding genomes. And there was actually an interesting puff piece on Craig Venter in the New York Times Magazine last week, which he says now that his group have got 2 candidates for parts-based minimal genomes that are in synthesis right now. So we might hear some interesting things from the Venter Institute by the end of the year, but if not, then let's assume they failed or again, they had a few errors like last time. So first on part of this, I'll just go through this quickly because starting to run out of time, was the DNA assembly. We wrote a review on the various methods of DNA assembly, and that was highlighted in a Nature Technology feature following on from the synthetic genome. We decided to follow up what we learned in our review with a semi-automated pipeline for our own DNA assembly. We can use extensive use of liquid handling robots, and really what I've been trying to get Arturo and James to focus on with this system is the ability to take any part and be able to place it in any position in the final construct and have it in any orientation. This is important to me because I'm really interested in understanding how the positions and orientations of parts that then make up a genome affect how that genome works. The system uses orthogonal linkers, and James McDonald has written a nice bit of in-house at the moment software. Hopefully we're going to spin out and have this on the web, which designs neutral orthogonal DNA sequence which can be used to link pieces together through the sort of overlap assembly methods, such as the way yeast puts things naturally together, or the way this Gibson assembly that everyone uses now puts things together. So we have a software system where you can define how long you want your linker to be, and it'll use various algorithms and plug into BLAST to try and make sure you end up with orthogonal linkers that have a reasonably neutral sequence and do not match anything seen in nature. So hopefully they encode for nothing except space. So this our first attempt at this DNA assembly has worked reasonably well. We can do combinatorial one-pot assembly of several parts. We can incorporate diversity for library synthesis within the process by doing mutagenic PCR of specific parts as the assembly is occurring. That's really nice. That means we can build something, and within the process one part becomes a library. We've really been looking at how the topological effects of putting genes in different positions comes into play. This small example here of a few genes that we've selected and rearranged on a plasmid. We've tried to insulate these genes from one another, and sure enough, the GFP and the RFP that are placed in different positions around this plasmid behave the same in all conditions. So that shows the context is not an issue in that system. So heading on now more towards the genome writing side of things, I have an undergraduate student who's taken on a really interesting project. My first sort of project running, where we take bioinformatics data and try and do something a bit akin to systems biology. And this one is trying to look at how much location matters. So if you're putting together a genome from parts, how the part will be affected by its position on the genome. So we can see the E. coli genome in this case. You have open reading frames all the way around on both strands, and there's all sorts of differences in terms of the way that the DNA will be treated at the replication origin compared to the replication termination region. On the flip side, most people are working with DNA characterization and creating their networks in E. coli on plasmids, which behave quite differently and will undoubtedly have different context to being on the genome itself. So let's say Druendi and their biofab can characterize 400 or 500 E. coli promoters. That would be quite useful and very accurate in terms of their expression levels, and then someone comes along and says, I want to build a 500 open reading frame genome, and I need this expression in all these pieces. It's not really sure that, because all that characterization of those promoters was done on plasmids, whether that would translate when they were put together on the genome. So to investigate whether this is true or not, we're just getting started here. It's just a snapshot of what we're doing. We've taken what they call the Zaslava collection, which is every genomic intergenic region between ORFs, that are effectively promoters, has been taken out by people at the Weizmann Institute, placed into a promoter, and then placed into a collection of cells and characterized with GFP output through things like growth from glucose as you move into lactose, and then you can monitor the behavior of all those different promoters, not on the genome, but how they behave at the plasmid level. And we found a rival collection, which is instead on the genome level, called the Taniguchi collection, which contains a nice amount of single cell reading data, where on the genome they've fused each gene to YFP. So the last lot was GFP on plasmids, and this time it's YFP on the genome. And we want to compare these YFP output values and GFP output values to see what the differences are, how many promoters sort of, I guess, play ball and behave exactly how they are on the genome as how they are on the plasmid, and how many say, nope, the rules don't apply to me. As soon as I'm on the plasmid, I behave completely different. So we have a whole lot of data available that we can mine for very similar growth conditions. There's existing microarray data, that's RNA levels from the genome. Now there's RNA-seq data to go with that, so that's a sort of upgraded version of the same sort of thing. There's the Taniguchi YFP proteomics data, and then there's the Zaslava collection, which is the sort of the standout one because this is coming from the plasmids instead. Oops. And this sort of talks us through the central dogma, and effectively by comparing all of these, we have reasonable measurements for all of these processes. So we compared experiments between microarrays, such as one done way back in about 2000 from Bernard Paulson's group with RNA-seq and RNA degradation, and YFP from the Taniguchi work, and the Zaslava data from Zaslava's papers. In the end, all of these data sets had some promoters missing, so in the end we only managed to get down to about 200 that we were confident are represented very well in all of these different data sets. And we can initially see very good correlations between some of the data sets. So the microarray data and RNA-seq data, which were done in completely different labs about 10 years apart, but in similar conditions, do show a reasonably good correlation here. So that at least is quite nice. That shows that comparable work done a long time apart still holds. And without going into much of the details, the conclusion so far is the YFP data has some reasonably good correlation with the RNA-seq microarray, RNA-seq times RNA deg data, but the real outlier is the Zaslava collection data, which correlates very poorly with almost everything. So it's sort of flagging up this warning that the plasmid-based characterization of parts is not, as you might imagine, is not very well representative of how those parts will behave if they're on a genome. And what we're interested in now and going to investigate further is which of these data points are the ones that behave. And we have, if I go back here, we have seen, for example, PTSG, this promoter here, behaves quite very similarly on the genome as it behaves in the Zaslava collection, has similar expression levels, and when you change the induction from glucose to glycerol, you get the same change. So this is an example of one of the promoters that behaves very well when you change it from genome to plasmid. And we have obviously numerous examples of promoters that completely behave differently. So moving on now, we also want to try this in yeast. Yeast is the organism that we've done most of our sort of apps-based synthetic biology work on. We'd like to see, we typically with the yeast apps, we integrate those into different into known positions within the yeast genome. And we do know from previous work from Jens Nielsen's group that the gene expression and regulation of promoters can be dramatically affected by the location within the Saccharomyces genome. And they've tested this with a couple of different promoters here, placing them in regions throughout the yeast genome, regions that they'd gone and researched as regions that they thought would be very neutral, but they actually still see significant differences. And what we've become really excited in is the synthetic yeast genome project, which Joel Bader will talk about in more detail later today, as that provides a sort of a very unique resource for investigating how context dependency, topology, and context rules affect gene expression levels. So just to give a plug for the synthetic yeast project, which Joel will talk about a bit more. That's Jeff Boker, Joel Bader, and colleagues at Johns Hopkins has kicked it off. It's now a major international project in 4 countries, and we've just got involved. Joel will probably go into this a bit more detail, but one of the real cool features of synthetic yeast, apart from opening up a 21st amino acid coding sequence, is it has this ability for automated refactoring of genomes, where these symmetrical LOXP sites are inserted in various positions along the genome so that you can induce recombination, and the genome effectively shuffles around, either inverting genes between the sites, excising them and losing them entirely, or translocating them for different positions. So what you effectively get is you've got a synthetic yeast which you can, through user induction, you can get it to shuffle its genome, and then you can grow it and select it in various conditions, and you can work out which of these shuffled genomes will survive and proliferate in those conditions. So it's a tool for working out the minimal genome, but also working out perhaps are there arrangements of the genome that are more favorable for certain conditions of growth. So for us, in terms of looking at genome context rules, that's fantastic. So we're proposing now chromosome 11 as the UK chromosome to synthesize the entire synthetic version of this chromosome as part of the synthetic yeast project. And the full UK contribution is proposed to be a bioresource so that we can make all the physical yeast strains and collections associated with this work and all the international work available for everyone to plug in and make use of, and also to teach the sort of genome engineering methods, expertise, and software that's required for this project to the community here. And we hope to run community workshops every year to help people learn how to do this kind of work, which there's a lot of interesting methods involved here where we're doing things such as creating 40 kb constructs in one go, and using all these various new DNA assembly technologies developed not just by this project but by Craig Venter's people to do this work. For me, this design rules for genome engineering is very interesting because there's a lot of potential applications about this, and talking with Anne in the last couple of years, one interesting one is the clustering of genes, and you can see that through evolution in yeast, you've got certain clusters have arisen, such as the DAL cluster, by shuffling the genes of this cluster from separate loci into one specific cluster. And we're interested in and have now kicked off a project to try and mimic that in synthetic yeast, where we're doing synthesis of the antibiotic penicillin, which has a history of being discovered at Imperial College. We have the genes now in plasmid form in our cells, and we have now this cute, very cool selection method where we co-culture our yeast against B. subtilis. We can nicely tune the amount of yeast in B. subtilis at the beginning of that co-culture, and it provides a continual pressure during a batch growth for the yeast to be producing penicillin at a continual rate to be able to kill off the B. subtilis. Without a continual presence of penicillin, the B. subtilis takes over and the yeast are killed off. They can't proliferate because the B. subtilis eats up all of the media. And the idea here is that this effectively, we want to be able to stick this pathway into a sort of synthetic subtelomeric region and use the tools of the synthetic yeast project, the recombination system, to be able to shuffle around the genes, their location, and perhaps have some of these genes in one subtelomere and one in a neighboring subtelomere, and see what we can shuffle around so that under this selective pressure that I described, we can use directed evolution to optimize the pathway in terms of its output. And perhaps then we can turn to the plant community and find other coding sequences we can throw into this to generate perhaps new antibiotics or new therapeutics that we can then also select for in a similar system. And we've already thought of an interesting acronym for this project. So we hope it goes somewhere, because I can't wait to tell this to more people. So this is my lab, and just to reiterate how much we love yeast, we went to Fuller's Brewery last year, and we found out that they really do have a centrifuge for spinning down vast amounts of E. coli and beer, and we told them about our flocculation project, and they weren't really sold on it because they're very traditional at Fuller's, and they're not really into synthetic biology beer. And finally, just to plug that this time next year, we're going to have the major synthetic biology meeting, SB6, at Imperial College, and we'll be putting out some calls for abstracts, etc., towards the end of this year. So please keep this time free in your diary if you're interested. Okay, thanks.